I'm going to start by explaining what the problem is we want to tackle with this research. So the subpopulation of autistic individuals with a co-occurring intellectual disability makes up 30% of the autistic population. They have significant limitation in social and cognitive functioning and also severe stereotyped and repetitive behaviors. Because of this aspect, it's difficult for these individuals to understand the outside world, and everyday stimuli like light or sound can be experienced as threatening or over-arousing. And this results in a massive amount of stress for these individuals, which is being contained by the expression of these stereotyped and repetitive behaviors like walking up and down the same way or making a sound over and over again. Despite their great need for interventions, the intervention in this population are lacking efficacy and are also often being excluded from scientific research. As pharmacological option in autism, the neuropeptide oxytocin is increasingly being studied. Oxytocin is produced by the hypothalamus and it acts both on the central and peripheric nervous system. It has social behavioral effects, but also emotional and stress regulatory effects. However, until now, research in autism mainly focused on social behavioral effects of oxytocin, lacking objective biophysiological markers of the stress regulatory effects of oxytocin. And also, until now, no study has been conducted investigating the stress regulatory effects of oxytocin in children with autism and an intellectual disability. So how do we want to help this population? It's by closing this research gap and investigating the stress regulatory effects of oxytocin in children with autism and an intellectual disability by use of two objective biophysiological markers. And we want to see if these physiological effects are also paired with behavioral effects. As first objective biophysiological markers, we're going to use cross-frequency coupling decoupling ratio of alpha and theta bands measured through EEG. So neural population fire at discrete frequency bands known as alpha and theta and so on, as you know. And these frequency bands, this oscillation can synchronize. As you can see on the screen, the alpha and theta excitatory phase meet at a ratio of 2:1. This is called harmonic cross-frequency coupling and is mainly seen during periods of active neural processing. What also can happen is the formation of spurious background couplings, which is mostly seen during period of cognitive load or cognitive stress. On the other hand, desynchronization of the oscillation can also happen, as you can see on the screen, by the maximum desynchronization state of alpha and theta, which calculated corresponds to a ratio of 1.6. And this is called non-harmonic cross-frequency decoupling, and is mostly seen during period of rest, like a meditative state. Also seen during meditation is that the formation of spurious background couplings is decreased. So the excitatory inhibitory imbalance model of autism posits that autistic individuals have an increased neural noise, and this can be interpreted as an increase in the spurious noisy background couplings and a decrease in the decoupled state, so a decreased signal-to-noise ratio. Oxytocin may mitigate this imbalance by increasing the decoupled state and decreasing the spurious noisy background couplings. And this is also something we saw in our previous study with children with autism and a normal IQ. We saw after a 4-week intervention with oxytocin that a significant increase of the decoupled state and a significant decrease of the spurious background coupling was seen in the oxytocin group and not in the placebo group. And it's also something we hypothesize we will see with the children with intellectual disability because of their severe basal stress. Our second objective biophysiological marker is the amount of autonomic cardiac arousal measured through high-frequency heart rate variability, which is assessed through ECG. So high-frequency heart rate variability can be seen as a pure measure of parasympathetic activity, as it represents the respiration-linked changes in heart rate and is widely accepted as a measurement of respiratory sinus arrhythmia. And decreased high-frequency heart rate variability is undesirable, as it represents the inadaptability of the heart to correctly respond to a stressor. And so oxytocin may mimic the effect of endogenous oxytocin by regulating the cardiac vagal system and increasing high-frequency heart rate variability, thereby stabilizing the cardiac autonomic arousal seen in autistic individuals, which is also something we saw in our previous study with children with autism and a normal IQ. At post-intervention, we saw a significant increase in high-frequency heart rate variability in the oxytocin group and not in the placebo group. And it's also something we hypothesize we will see with the children with intellectual disability. So how do we plan to accomplish all of this? We have started a double-blind, placebo-controlled, randomized clinical trial where we are recruiting 80 children aged 4 to 13 years old with a diagnosis of autism and intellectual disability. We will have an intervention of 4 weeks, 24 international units of oxytocin or placebo given 3 times a week. We will have measurement of the biophysiological markers and behavioral assessment at baseline, post-intervention, and at a 1-month follow-up. Both EEG and ECG measurements are taken during a 7-minute resting state, where the children are presented with a movie paradigm called Inscapes that you can see on the screen right now. This movie paradigm is made so that it has no narrative or no scene cuts, so it increases compliance from the children while minimalizing cognitive load. So thank you for your attention.